Because there are notable differences in the size of molecules, chemical and enzymatic stability and cell concentration are required certain precautions in the extraction of nucleic acids. For example, the DNA requires soft procedures to avoid its fragmentation. In the case of RNA, installation must be done very carefully to avoid the action of enzymes degrading as RNA. Depending on the biological source, which will extract the genetic material as well as its cellular location, since it can be that the procedures become as simple as removing genetic material from bacteria culture or more complex such as suspension of virus where the DNA is associated with capsular proteins. According to the next chart, depending on the organism we want to extract nucleic acids, are the solutions and concentrations we need to use and to prepare the cell for the extraction.
For the samples of DNA and RNA, a dilution of 100 was made of both samples, and after we took 1 milliliter of each one and put it in an spectrophotometric cell. To prepare the DNA for making electrophoresis, you have to centrifuge to obtain the pellet that have the leukocytes. Then, sodium acetate and proteinase was added and the pellet was resuspended with the vortex. Then, isoamylic phenyl chloroform alcohol to denaturalize the peptides. Finally, a wash of ethanol was made. To prepare RNA, you have to centrifuge and add lysis buffer to the pellet. Chloroform was added and we centrifuged again. After, we added ethanol at 70% to make the wash. The lectures obtained with the spectrophotometer were 2060 and 2080. The concentration of DNA was 0.3667 micrograms per milliliter, and the final amount was 1.1001. The concentration of RNA was 10.58 micrograms per milliliter, and the amount was 42.32 micrograms. To make the electrophoresis, 5 microliters of each sample was used. The first lane had the oxygen marker. On the second and fourth lane, the sample used is the same. In the second was used the bottom part of the microtube, and in the fourth was used the supernodant of the same microtube. And in the third was used RNA sample. All the samples have contamination with proteins. In RNA samples, it's difficult to see 3 bands because we have higher concentration of RNA. Also, have contamination with genomic DNA. In lane 2 and 4, we can see genomic DNA.
In our sample, in the case of the spectrophotometry, in the comparison between 8260 and 8280 from the DNA is 0.7917. It means that it's pretty pure because a value of 1.7 means 100% pure and a value of 0.5 means 100% of contamination with proteins, so our result is more close to the second one, so it's contaminated. And in the case of RNA, our value is 1.2532. In this case, the best value is 2, and a value lower than 2 means the sample is contaminated with proteins, so this value is caused by a residue of phenol in the sample. There are a few differences in the protocols of DNA and RNA. One of the differences is that the DNA was incubated 1 hour at 37 Celsius degrees. The RNA you have to use cells to buffer to lysis the cell, and the time of incubation are 10 minutes at the same temperature. And in the RNA, you have to add 3 cells of the purification. These are only a few differences between DNA and RNA protocols.
Many problems could present when we're doing a PCR and we have a contaminated sample. For example, if the sample we're going to use is from DNA and it doesn't have an appropriate purification level, PCR won't be properly done. And this doesn't affect only for DNA, also for RNA samples. When we have our sample contaminated, we can also see that the PCR will have some degradation, or maybe that the polymerase will inhibit sequenciation. It depends on the kind of contamination. So we should be careful of trying to have a sample free of nucleases, RNases, DNases in each case, or salts, or even ethanol. By this way, we are going to avoid these kind of problems when running a PCR. In the second lane where the DNA was run, we can see a very clear band. Why is it that clear? Well, the answer may be that it was a very low concentration of our sample. This can also be compared with the numbers that we got in the spectrophotometer. In the spectrophotometer, we got a concentration of 0.3667 micrograms over milliliter. This is a very low number according to what literature wants us to have, like 1.7. So maybe it was a very low concentration, and that's why we got a very clear band. In the other hand, in the lane where we have RNA, we can see a very big dot. Why? Well, this can be explained because maybe we got a very high concentration in the sample, and this can be also compared with the spectrophotometer. In the spectrophotometer, we got a concentration of 10.58 micrograms over milliliter. This can also explain why we got these results in the PCR. And even if we're noticing the concentration of sample in each of the lanes, we also need to see that we have some contamination. For example, in RNA's lane, we doesn't have only the RNA part, we also have a little contamination with DNA. This is by the band that we have at the height of the DNA markers that we got in the second and fourth lane. And it's also important to see and notice that we have nucleus contamination in each of the pits, in the pit 2, 3, and 4. So we doesn't only have different concentrations of samples, we also have contamination, and this is something that we should correct in the future.
In conclusion, RNA or DNA samples should be treated for purification in a different way because of its biochemical characteristics. On the other hand, it is important to be precise measurement of each of the compounds added to nucleic acid samples, since a mistake can cause fermentation or degradation. In addition to methods of quantification, the use of alarm gel allows to observe more precise characteristics of the extraction of nucleic acids as the existence of contamination in the sample. It is important to emphasize the voltage and the percentage of alarms for electrophoresis that were appropriate for which samples were clearly observed.